Thanks. First, I would like to thank the organizers for selecting my talk today for this Usher Symposium. As you all know now, the majority of the genes that are involved in Usher syndrome are mainly large genes. So there is a need to extend gene therapy by developing gene editing strategies for this type of disease. And this will require adequate delivery tools in order to deliver efficiently and safely these strategies. So today I will present new non-viral vectors to deliver directly the Cas9 as a protein into the retinal cells. So what is the state of the art for retinal degeneration? First, you have the gene replacement strategies that work really well in photoreceptors, with the first landmark Luxturna product, which consists of bringing a healthy copy of the RPE65 gene into the cells. And luckily, these genes fit into one AAV, which is the only viral vectors that can efficiently transduce the photoreceptors. So this healthy copy will compensate the function of the endogenous mutated genes. However, this type of strategy can be applied only for monogenic recessive disease due to mutations that are in small genes. So what can we do for large gene? We can use gene editing strategies, and indeed there is a first clinical trial ongoing right now that consists of bringing one Cas9 and two guide RNAs to remove cryptic splicing sites in the endogenous gene. So you have this first strategy ongoing in clinics, and you can also use other gene editing strategies. The most easy one is to have one Cas9 and one guide RNA. This will lead to a double-stranding break that will be repaired by the cells with the NHEG pathway in non-dividing cells, and will lead in insertion and deletion, so indels and the gene knockdown. But this strategy cannot be applied for all the disease. And in fact, there is new editors, base editing and prime editing, that can correct precisely mutations and can correct without double-stranding break the vast majority of the mutations that lead to inherited retinal disease. So as you can see, it's promising, but the question is how to deliver them in vivo into the photoreceptors. So you could deliver the DNA thanks to an AAV, but there is some limitations due to the limited packaging capacity of the AAV, and also this will lead to the long-term exposures of the Cas9 that can increase the off-targets and the immune responses. So in fact, the most straightforward approach should be to deliver directly the Cas9 protein with its guide RNA. This will allow a transient delivery without the need of a transcription or a translation steps. And this has been already applied successfully in the RPE and in the inner ear, and so it never be applied in the photoreceptors. That's what we want to try. So to do so, you need to take into account all the obstacles that is specific to the retina. So after a subretinal injection, the Cas9 protein will have to overcome external limiting membrane, the extracellular matrix, and you don't need to forget the external segment of the photoreceptors that are quite dense. And still, the Cas9 protein will have to cross the cell membrane and escape from the endosome to finally enter to the nucleus. So we decided to have a proof of concept to first take the system, the most simple system, which is the Cas9 protein, which is quite a big protein, and one guide RNA, which is highly negatively charged. And this form a ribonucleoprotein complex, the RNP, which is negatively charged and normally should not cross the cell membrane. But based on a previous study done in the brain, we know that the nuclear localization signals that are in the Cas9 protein, in the periphery of the Cas9 protein, and that are positively charged, can mediate the RNP entry in neuronal cells. So we wanted to try that in vivo in wild-type adult mice, and we analyzed the percentage of indel formation 7 days later in the whole RPE and in the whole retina. So as you can see, we were able to reach up to 4% of indels in the RPE for the highest concentrations. And in the retina, for the first time, we showed that it is possible to edit the retinal cells. But you see, we achieve up to 0.8%. So it's quite a low percentage of indels. And we wanted to improve this by adding the RNP with non-viral vectors. So in fact, there is different approaches for RNP delivery in vivo, and we wanted to focus on chemical methods and more complexation methods. So complexation is really a balance between cationic and anionic charge. And we decided to focus more on cationic lipids and try also cell-penetrating peptides, so CPP, which is a small polycationic peptide that can facilitate the entry of the RNP. And finally, we tried also targeting peptide that can direct the RNP into a specific cell type, so the photoreceptors for us. So first, we tried cationic lipids because it has been already shown to successfully mediate the RNP entry in the inner ear and in the RPE. And we complex it with cationic lipids commercially available. And to localize the protein Cas9 in the retina, we decided to link it with cyanide-free fluorochrome. And so 3 days after the injection, we showed that, in fact, the Cas9 protein is mainly in the RPE, and we think it's maybe due to the phagocytosis capacity of the RPE. And it seems that the cationic lipid doesn't mediate efficient Cas9 protein delivery into the photoreceptor nuclei. So as you can see, in fact, there is a lot of large aggregates in the subretinal space. So we thought that the Cas9 lipids form with the RNP big complex, and we wanted to go to another cationic vectors, much more smaller. So we wanted to try a lipopeptide, which is composed of a small lipid tail, a cell-penetrating peptide, and a targeting peptide. And this lipopeptide can form particles of 80 nanometers, so more smaller, and can complex efficiently the RNP. And we decided to change the targeting peptide, which was initially target the tumor cells, to a sequence that we know specific for retinal targeting. But as you can see, in fact, it doesn't change anything, and we still have a low percentage of indels in the retina. So we think that this is maybe due to the lipids. The lipids is possibly the problem, and we decided to go for a fully peptidic system. So we keep the CPP, and we linked it with an endosomolytic peptide, which can be facilitate the endosomal escape, which is a big barrier for non-viral vectors. And this shuttle peptide has been it has been shown that it can efficiently complex the RNP and mediate the RNP delivery into epithelial cells. Sorry. So as you can see, after subretinal injection, after 7 days, we were able to reach 4% of indel in the RPE and 0.8 in the retina. So still quite the same as what we can achieve with the Cas9 protein alone. So to go further, we decided to change, in fact, the CPP with this red sequence. And for this experiment, we performed a 1 microliter subrachional injection. And after 7 days, we can see that this shuttle peptide, with this red targeting peptide, can increase significantly compared to the initial shuttle peptide, the percentage of indels in the retina, and it was not the case in the RP. So to conclude, we really wanted to try different categories of non-viral vectors to deliver the Cas9 protein into the photoreceptors. And the peptide and the targeting peptide seems to be a good candidate. And what we want to do next is to link covalently this peptide to the Cas9 protein to have stable, more stable and more small complex in order to increase the delivery in the photoreceptors. And of course, in the context of Usher syndrome, it would be really interesting to try this strategy in the inner ear. So I would like to thank all the IDV, all the members of the team 15, my PI Denise Dalcara, and Juliette Pullman, who's working also with me on this project, and our collaborators.